We're going to be covering material in that exam, starting with measurement issues and going through this segment on steroidogenesis. So I'm going to finish off that material in the first few minutes today. And then we're going to start talking about sex. Meiosis, gametogenesis, in both genders, and get into the segment on reproduction. That will not be on the exam today, not today, on Friday. Okay. All right. I showed you this illustration the other day. What is it? Any ideas what that picture is? Okay. It's a diagram that attempts to capture the salient features of receptors for most steroids and for similar ligands or similar similar molecules like thyroid hormone, vitamin D, retinoic acids, things that tend to be lipophilic, that have nuclear type receptors. And when you look at those, they all seem to share some structural features which are on this image. So they have essentially a three-part structure, DNA binding in the middle, that is mediated by these two zinc fingers and the associated nucleotides, amino acids around them, which interact with nucleotides in DNA. They're held together by cysteine groups. And there are other portions of these protein molecules that are involved in transcription or in binding to the hormone, in dimerization, which I'll get to in a bit, interaction with other receptor molecules, and transcriptional activation as well. So that structure, remember, it covers a whole family of receptors at the nuclear level. The DNA here is shown in green here. This is what glucocorticoid receptors would look like when they're complexed with DNA. This is a stereo pair again. I've shown you these at least once or twice. This is a stereo pair. If you cross your eyes and relax them, you can see a three-dimensional image of the DNA associated with the protein. The one thing to note about the glucocorticoid receptors here is that in each of these images, there's actually two of those molecules. There's one down here, and there's one up there. And they are oriented head to tail. They're like taking your two hands and locking them together. Okay. They form what's called a homodimer. All of those receptors I showed you in the last slide have a tendency to bind DNA as homodimers. They can also form heterodimers. So one half of the pair can be a glucocorticoid receptor. The other half could be a vitamin D receptor. So you can get heteronuclear receptor binding to sites on DNA so that they only turn on in the presence, in that case, of glucocorticoids and vitamin A. Vitamin D also has a receptor like this. Here's another picture showing you cyclic AMP receptor binding protein, or CREB. Remember, that only works when it's phosphorylated. And here are two molecules of the CREB binding to DNA. And the one thing to note here is that when they bind, they bend the DNA. And that bending of DNA tends to make it more open to transcription. So there's a physical, there's actual physical change in the DNA that occurs on binding of some of these hormone receptor complexes that can help describe how the hormones are actually altering transcription. Okay, yeah. So that's nuclear receptors. The last thing I want to talk about steroid metabolism, steroidogenesis, is not the synthesis of the molecules, but their breakdown. The other end of the chain, if it as you were. Steroids would be normally found in circulation, other than those that are obtained in the diet and come in through the stomach. In either way, they're going to pass through the upper intestine. And when they pass through the upper intestine, they come into contact with the products of the gallbladder. And those products tend to be cholic acids. They're detergent-like molecules that help solubilize the lipids that are in the stomach contents as they pass through the intestine, makes them more available to be absorbed, as, for instance, chylomicrons. So this mixture of contents of the stomach, contents of the gallbladder, and there's one other thing that's not shown here. It's the contents of the apocrine portion, that is the non-endocrine portion of the pancreas. And that includes bicarbonate, lots and lots of bicarbonate, along with proteolytic, nucleolytic, lipolytic enzymes. And those get dumped in at about the same place, changes the pH. Remember, this is quite acid. Changes the pH of the contents from the stomach, forms these micelles, makes things more absorbable in the small intestine. As it's doing that, if you've got steroids coming in, those steroids are going to be absorbed into circulation, and they'll often find their way to the liver, often on the first time through circulation. And when they do, the liver is going to make modifications such as inserting OH groups, double bonds sometimes, and conjugating them to sugars or sulfate groups that make these molecules more water soluble. A lot of them end up going into the contents of the gallbladder. And so they find their way back into the gut. And they may pass all the way through, but they may also end up going back through and having another pass. If they're really water soluble, they may get filtered by the glomerular apparatus into the incipient urine and get passed out as waste as part of the urine. So this cycle of uptake into blood, passage through the liver, hydroxylation or modification to make them more water soluble, and then back, usually via the gallbladder, into the intestine. This is called enterohepatic circulation. Entero, the entero system, the gut system, hepatic, liver. So this is enterohepatic circulation. And it involves what are called phase 1 and phase 2 enzymes. The phase 1 enzymes are largely those involved in introducing water, modifying the molecules in other ways, adding or breaking double bonds. Type 2 or phase 2 enzymes are those such as sulfatases, sulfurolases, which add sulfates, and glucuronidases, which glucuronyl transferases, I should say, which add glucuronic acid in similar molecules. So phase 1 and phase 2. All lipophilic compounds will have some form of enterohepatic circulation. So steroids do this. Many drugs, which are lipophilic, will undergo this circulation. The breakdown products of hemoglobin, the bilirubins, will go through this cycle. Toxins that you may find in animal or plant products will go through this cycle. So this is a very important cycle. You can imagine the situation or imagine some of what could happen if you introduced a lot of steroid or a lot of drug or a lot of lipophilic material generally, and then you added normal hormone levels on top of that. Okay. If you read the package on a lot of drugs, okay, say for instance antibiotics, it will tell you that if you're taking contraceptive steroids, you should talk to your doctor. Any idea why? Any suggestion of what might be going on there? Any guess? Hint, hint.
Can it make the contraceptive less effective?
Will it may make the contraceptive less effective, but in that instance, it's on the erythromycin or on the streptomycin. It's on the antibiotic. So what's being more affected in this instance? The antibiotic, okay? So if you have already loaded the system with drugs or steroids that are being metabolized in this pathway, and you add something to it, that may be metabolized in a different way. There could be competition, which would make it stick around longer. Okay? Or that pre-treatment may have amped up the phase 1 or phase 2 enzyme so it works faster. So it would clear the drug more rapidly. You'll see all sorts of those potential conflicts. Has anybody got elder relatives who are taking 5 or 6 or 7 medications on a daily basis? Okay. Or even relatives who are being treated for cancer or something like that. So they got 5 or 6 different drugs, right? Has anybody talked to a pharmacist about the interactions between those drugs? They should. And this is primarily why. Because they may interfere with one another in terms of their actions, in part because of the way they get broken down, because they share pathways for breaking them down. This is essentially the same picture, but it's drawn regarding biliverdin and bilirubin, the breakdown products of heme, from hemoglobin, red blood cells. And it's showing you the passage, the enterohepatic circulation, and showing you that about 90% of these bile salts are reabsorbed in the ileum as urobilinogen, and about 10% are lost in the feces. So there's a lot of breakdown going on. Okay, I think that this was just a reminder indicating that a lot of these hormonal pathways, the steroid-involving pathways, also interact with pathways like prolactin. So here, estradiol. Estradiol, given on a long-term basis, will actually stimulate the proliferation of lactotropes. So women who have been pregnant actually have more lactotropes at the end of pregnancy than they had at the beginning. Women who have been on long-term steroidal contraceptives have an increase in the number of lactotropes present after that treatment than they had at the beginning. Men who would be taking testosterone replacement may well modify the number of the lactotropes particularly, but potentially some of these other hypophysial hormones as well, because of the cell types. But you'll see interactions of all of these levels. Sometimes it's indirect via the binding proteins. For instance, in the thyroid system, thyroid binding globulin is under the influence of estrogen in terms of its production and androgen. Growth hormone binding proteins in circulation and IGF binding proteins in circulation are also sensitive to the circulating levels of steroids. Okay, so it's tied to a lot of different regulatory systems. Sex steroids are essential to maintaining the species, but for individuals, they're dispensable. Glucocorticoids and mineralocorticoids, on the other hand, are homeostatic and are absolutely essential for maintaining individual growth and development. And therefore, because they support life, they also support the population as well. Okay, and endocrinologists will always be interested in steroids, so are a lot of other people. I think that does it for that. All right. So everybody has this stuff memorized? Got it all down? That coat hanger that for all metabolites of C21? All right. All right. I have a question for you. Two words you come across in cell biology: mitosis, meiosis. What's mitosis? Somatic cell division. Okay. What does that mean? Any cell that's not a sex? Not a sex cell. What's a sex cell? Pardon? Are not part of the gonads? Anybody want to correct him?
They're secreted by the gonads. Gametes are secreted by the gonads.
Okay, they aren't secreted, they're generated. Okay? Mitosis is standard or replicative. Somatic cell division. What's meiosis? Somebody says gametogenesis. Is that correct? Okay. So, is it replicative?
Yes.
Yes.
Yes. But I thought this was replicative. Yeah. It's reproductive.
Reduction.
It's reductive.
Reduction of
Okay. So far I've got replicative. Reduction. Gametes. Something tells me that hasn't quite sunk in yet. Okay, what it actually is. Can this happen in plants?
Yes.
Sure can. What about fungi?
Yeah.
Yep. How about bacteria? Not sure. Can this happen in bacteria?
Yes.
Okay. So we're not sure if this can happen in bacteria. This happens in vertebrates, definitely. Let me come back to the gonads. The gonads only have gamete cells,
No.
ovaries and testes.
No.
Do they have some somatic cells?
Yes.
Yeah, in fact, most of what the substance of those tissues are somatic cells, which undergo mitotic division. Okay. So meiosis is cell division leading to production of gametes. There are two stages. How many stages are there in mitosis? It's one. Okay. During that one stage, you do what to the DNA? You replicate it, and then what happens?
double.
Okay. You pass a single copy of the whole set of chromosomes to one daughter cell, and the rest of it, remaining portion, hopefully the same kind of stuff goes into the other daughter cell. So you get DNA, replication, and equal division into identical daughter cells. Why didn't they call them son cells? Nomenclature. You get cell division leading to production of gametes. There's two stages. Stage one is DNA division, or replication, pardon me. And cell division. What's stage two? Stage two. What happens in stage two meiosis?
Reduction of
Okay, you form, okay, so the daughter cells don't just stay like they are down here, but they continue on to undergo a reductive division. So this is reductive. And although we talk about 2 stages, this is one process. It doesn't just stop in the middle. Although I'm going to show you, in fact, that a halting of this process is very important in distinguishing what happens in the male and the female. Okay, so what's on the slide? Okay. So, cell division, mitosis, replicative simple cell division, interphase, prophase, metaphase, anaphase, telophase. I suppose you can each and every one of you identify those under a microscope. Yes? Okay, you saw them in onion root tip cells. In biology 111, you probably saw something like it in high school. Okay, their interphase, okay, you start out at 2N in terms of the copy number of chromosomes. You get DNA synthesis, nuclear envelope breakdown, replication of the centrioles that move to the poles, and the beginning of the spindle formation. In metaphase, you've got the alignment of all the chromosomes in the metaphase plate, and there's 4N copy number of the DNA of the chromosome. At anaphase, you start to have these move apart toward the poles, and then finally in telophase, you actually get separation in cytokinesis, or breaking into 2 daughter cells. So, yeah, it's a complicated process, but it's replicative and relatively simple. Meiosis, 2 stages, the replicative division. You start out with 2N. These are would be the germ stem cells. And in the case of the female, you would have oogonia. In the male, it's spermatogonia. When they commit to meiosis, they undergo replicative division. They go through an interphase where DNA synthesis, and this is the big difference, crossing over occurs. What's crossing over? Hang on. I'm going to go to somebody else. What is crossing over?
Exchange of materials.
Pardon?
Exchange of materials.
Exchange of materials. What do we mean by exchange of materials? Okay, genetic material. So what's happening to that genetic material? It's recombining in different ways. What's the advantage to doing that?
Variation.
Pardon?
Genetic variation.
Genetic variation. What good is genetic variation? I want my daughter to be exactly like me. Well, maybe not quite exactly like me. Certainly my son needs to be exactly like me, right?
Can produce an offspring with better fitness?
Okay. Produce an offspring with better fitness. That sounds heretical. What is he espousing? Pardon? Mendelian genetics. What's the basis for Mendelian genetics, however? We found out after Mendel, but what's the basis for Mendelian genetics? Evolution. How do we find out about evolution?
Natural
Ah, who's this? Charles Darwin. Who is he? Your uncle? Okay, tell me more about your uncle.
He came up with a theory of evolution.
He came up with a theory of evolution. Was he the only one?
No.
Who was the other guy?
Someone's uncle.
There was one other guy who came up with essentially the same idea.
Less famous.
Pardon?
Less famous.
The less famous Darwin. Okay? It's a line off of the coast of Australia, between Australia and no, not the Galapagos, but the Malay Peninsula and the islands off the Malay Peninsula. Look it up. Okay. Okay. So, Charles Darwin, theory of selection, selection of the fittest based on genetic recombinations that Mendel started to show us about, but that we now teased apart in molecular detail and know a great deal about. Crossing over is the natural occurrence of DNA assortment. The assortment of, it's not just genes, it's often parts of genes. Okay. So it's particular motifs in proteins that get swapped from one gene to another that ultimately make the cell and the organism more fit. Okay. Any of you working in labs now? Gesundheit, by the way. Okay, you're working in labs. Do you play with this
Yes.
by artificial means? How do you do it? Transgenic mice. Okay, what are you doing with the transgenic mice?
Are introducing an exogenous gene into a gene of interest so that both are expressed under controlled circumstances.
Okay, and how do you do it? Do you just mate mice?
No,
What do you do first?
you have to inject the gene in a viral vector into the stem cell or something, I forgot, something, so that you have a homozygous transgene and then you breed it with a wild type mouse so that you end up with a heterozygous
Okay.
transgenic mouse.
So you take early embryos, and we'll talk about this in probably a week or so, and you introduce a vector, or you can do it with modified stem cells. You can actually take the stem cells and inject them into the embryos. You introduce that back into a female's reproductive tract and allow development of those organisms, which you then can mate back to others to proliferate your construct. Is there any other way that it's being played with?
So we do like PCR mediated transformations,
Yeah?
and then so yeast can like mitotically divide as haploids, so their life cycle is a little different.
Mm-hmm.
But once you've mutated, say, a haploid strain, you're going to want to mate it to a wild type.
Okay.
And then in order to get a homozygote, you would sporulate
Form gametes.
and form the gametes, and then mate to the two different sex types, essentially, to get a homozygous diploid. Because
Okay.
when they mate to form a diploid, they just combine all the genetic contents.
Anybody here have done any or read anything about something called CRISPR-Cas? Okay. What? What's it used for?
It's used for bacteria can use it to, or I can use it to basically get the genetic information of the virus or something that might hurt them.
Okay.
And use that to find the virus to attack it later on, or there are other uses for it too.
Okay. Okay, it's being used in laboratories to essentially do what crossing over does in meiosis. You are taking segments of DNA from one background and swapping them into another. Okay. We now have multiple ways of doing this, but that's one of them. And it can be done in bacteria, it can be done in yeast, it can be done in virtually any organism at this point. And in fact, this spring and summer, there have been some transgenesis well, they're still experiments, but attempts to use transgenesis in treatment of humans over in this country over this past summer. And I forget what the disease they were treating. But it's gotten to the point where we're manipulating DNA, even in humans, using some of these tools. But the whole thing started with beginning to understand what crossing over was occurring in meiosis, okay, in that first stage, that first meiotic stage. Normally you get prophase, metaphase, anaphase, telophase. So essentially replicate looks a lot like mitosis. You end up with two cells that are 2N at the end, just like you would in mitosis. But rather than stopping here, they immediately enter the second stage of reductive division. And rather than 2N cells as a product, you end up with 4 1N cells as product. Now, are the products of meiosis the same as the products, genetically the same as the products you get in mitosis, being identical?
No.
No. So you can get 4 different combinations of genes in those gametes. So non-identical daughter or son cells. One other comment I want to make. This stage turns out to be approximately the stage at which oogenesis halts in mammals. And it may halt for 50 years. Okay. Not necessarily a short period of time. That doesn't occur in spermatogenesis. Some of these next slides, I'm not going to go through in detail. What they are showing you the various substages in meiosis. So there's a leptotene, a zygotene, a pachytene, a diplotene, and a diakinetic stage in the meiosis prophase 1. And the diplotene stage is the state where oocytes tend to halt for prolonged periods of time. We'll get to why they tend to halt, or what we think is the reason they halt, in the next lecture or two. So this is just, again, showing the stages of meiosis: metaphase 1, anaphase 1, telophase 1, and then entering meiosis 2, where you get prophase 2, and metaphase 2, anaphase 2, and finally cytokinesis to form the gametes. One thing that happens in spermatogenesis that's not shown here on this diagram is you sometimes maintain little cytoplasmic bridges between the cellular products in gamete formation. And that can mean, if there's a problem with one of these, that information associated with forcing this to go toward apoptosis gets promulgated through the whole series. So you, rather than losing just one cell, you often lose 4 under those types of circumstances. You won't find that happening in oocyte production. All right. Now I go here. Go to here. Oh, steroid chemistry, that's the wrong one. Take that one. So I'm going to go back to. When we get into gametogenesis, we're going to be working from a lot of the slides that are shown in the PowerPoints on male and female endocrinology. So, I'll begin the process, or this, we've talked a little bit of meiosis. The whole, when I talk about reproduction in the male, there'll be a similar outline for the female. I'll go over the anatomy. I will eventually go over the embryogenesis and the sex determination. We've already gone over a lot of the hormonal controls. We'll talk about puberty after we talk about pregnancy. We'll get to the testicular anatomy, some on meiosis. In fact, I'm going to go to these slides first today, and then we'll talk more specifically about spermatogenesis and more on contraception and aging. And we'll do that for the female as well. Over here, I want to go down to okay. Now, you know it's complicated enough understanding the difference between mitosis and meiosis. Well, you're going to be upset with me when I start telling you that meiosis comes in multiple flavors. In vertebrates, there's a version of meiosis that occurs in males, and there's a more complicated version that occurs in females. So I keep telling you males are simple. And here again, we have another instance where males are more simple than females. And under these circumstances, the male meiosis starts with a parent cell. This would be a spermatogonium. And it goes through prophase, metaphase, where you've got DNA replication, you've got crossing over occurring, giving you hybrid chromosomes. You end up with a metaphase 1 giving assorted genetic material to two daughter cells. And those two daughter cells then go on immediately to form 4 daughter cells that in this case are genetically not identical. So each one of these, if it were to differentiate fully, would become an individual sperm with an individual sperm's complement, one end complement of chromosomes, and slightly different alleles among various genes that it encodes for. But that's a fairly simple, straightforward picture for meiosis. The female, different, particularly in mammals, And in mammals, you start out somewhat the same. You get initial multiplication of the primordial germ cells by mitosis. That occurs in the males as well. So the oogonia in this case, prior to committing to meiosis, are dividing mitotically. And this is well prior to birth. In males, the spermatogonia, before they commit to meiosis, also divide mitotically. And in males, that mitosis can continue to occur after birth, through adulthood, and essentially until death. So replication of these parent cells in the male continue for the entire life of the individual. In the female, this mitotic replication of oogonia stops prior to birth. And they will migrate, they continue to mitose during this migration to the genital ridge, which is going to become the ovary in this case. And you form these oogonia, as I said, stop replication, mitotic replication, before birth normally, or just about the time of birth in a few species. They will enter a final interphase of meiosis 1. So they're committing, when they finally commit to meiosis, they'll enter the interphase and synthesize DNA. You get crossing over occurring, and then you stop, you halt the progression of meiosis 1 prior to birth in most animals, and you don't resume it again until the conditions are appropriate, both hormonally, nutritionally, and developmentally, for that oocyte to resume growth. Up until between the time of birth and the time of puberty, all of those oocytes that may commit or may move beyond this halting point will be lost. You won't see those being used for production of oocytes. But once you hit puberty and the hormonal conditions are right for development of the oocyte, you will start to see oocyte meiosis resumed, and at about the time of ovulation, you're going to see the production of or the completion of the first meiotic division to give you an immature oocyte and what's called here a secondary oocyte or polar body. So this is 2N, that's 2N at this point. And it's not completed. Meiosis isn't completed until contact with sperm triggers a calcium flux that causes resumption of meiosis, extrusion of a second polar body, this time 1N, and acceptance of the sperm pronucleus into the egg, which has now got a female and a male pronucleus. And then you'll get fertilization. But meiosis in the female, very different from meiosis in the male. Okay, I will see you folks all